So the first lab tip is basically around drawing our scientific diagrams. So we all had a chance to take a look at the mitosis root tips, and just wanted to highlight a couple things about this. So this is probably what you saw, something very similar to this. This was an image that I took through the microscope lens. You can see some of the cells, different stages of mitosis. I'm seeing some metaphase kind of near the bottom there. So right here, and I don't know if I see any of the other stages. Most of these other ones look like they are in interphase, so maybe I didn't go far enough down. Here is the scientific name for onion, so you're going to need that at some point. And so what I have here are a couple of diagrams, right? In terms of making a scientific diagram. So I'm asking which is better. Hopefully it's pretty obvious in this case which one is better. So if you take a look, the one on the left is larger. You can see a little bit more detail in it. It's more carefully constructed. All the labels are on the right side. I used a ruler to point to the cell part, and there's no shading or coloring or anything like that. The one on the right you can see is just on several levels just isn't quite as nice. So your diagram might look something like that, or if you're looking at a different stage of mitosis, maybe you're showing the chromosomes in a little bit of a different diagram, but probably not much different than something like that. The whole point of this exercise was mostly just to get you familiar with the idea of scientific drawings and to start to think about the other concepts around it. So learning a little bit about having a proper title. So you're going to have your binomial name. That was on the previous slide. Remember, it's Allium cepa, I think. And also really just those calculations in terms of determining the size of the specimen and then the magnification of your drawing. So that's what I'm looking for that particular part of the lab report. I'll address some other parts in future lectures. Back to topic 2. Topic 2, we covered lipids, we covered carbohydrates. We were making some notes about them. I'll come back to the Word document with the notes in a couple of minutes. Now we're going to talk about proteins. So proteins are really important. They do all sorts of things in the cell, and they're actually a big part of the human body. You can see it says they're 50% of the dry weight of the cell. In terms of the human body, proteins are, I think they're around 70% of the dry weight of the cell. So that dry weight means, of course, no water, right? Because water is a big chunk of our body. So what do proteins do? So proteins are, many of them are enzymes. Enzymes, of course, catalyze biological reactions, and we'll talk about them more in topic I think it's topic 9. We're going to talk about enzymes in more detail. But there's many other types of proteins out there, and kind of just go through some of the functions here, and then I'll write some of them down in the notes there. Some proteins are defensive. There's a lot of talk about antibodies lately in terms of what our immune system is producing from getting the vaccines. Some proteins are involved in storage of energy or nutrients. So ovalbumin is a storage protein that releases amino acids as a chick embryo is developing in a chicken egg. Some proteins are transport proteins. We're going to talk about them in topic 4 when we talk about membranes, and we have these proteins that are allowing solutes to be transported across membranes. Many functions for proteins. Some are hormones, such as insulin. Some are involved in message transmission between cells. You can see there's a neuron and some sort of receptor tissue, maybe muscle tissue, and proteins are involved in those reception processes. Of course, a big one, if you're an animal, is that proteins are involved in your muscle tissue. So actin and myosin are proteins. And then there's a lot of structural proteins in the body, collagen and keratin and things like that. Collagen is found in your skin. It's kind of a rope-like protein, and giving your skin a little bit of flexibility and bounce. So the next question, of course, is what are proteins made of? Proteins are made of a type of molecule called an amino acid. I didn't bring my pen today, so I'm just going to move my cursor and show you a couple of things here, and maybe at some point I'll I'll draw an amino acid for you in class when we talk about proteins later on in the semester. Oops, I did not mean to do that. Let me go back up here. So an amino acid, I just want to point out a couple of things. I'm looking at this one here in the middle, valine. So notice valine has the C double bond O, C negative. That's a carboxylic acid group. That's actually a carboxylate ion. It's the hydrogen has been removed, but it's basically an acid group there. And this group over here is called an amino group. So you can see where it gets its name, amino acid. It has an amino group and it has an acid group on it. So notice that compared to fats and carbohydrates, now we're looking at different functional groups and we have nitrogens. So that's a big difference between fats and carbohydrates and proteins. Proteins have these amino groups which have nitrogens in them. And they're loaded with nitrogens, one for every amino acid. Something about the amino acids is that there are 20 different ones used biologically. You can see some of them here. And if you take a look at the yellow structure on each of these proteins, it's different, right? So we have valine has 3 carbons, alanine here on the left has 1, glycine has no carbons. This group sometimes is called the side chain, meaning it's different for every amino acid. And the ones we're currently looking at are all hydrophobic side chains. So it turns out that the amino acids can have different chemical properties, which makes proteins very versatile. All the amino acids, by the way, have a name, so like valine, and then they have a 3-letter code VAL and a 1-letter code VAL or V, for example. If you have hydrophobic amino acids, you're going to have hydrophilic ones. So all of these have polar groups. Most of them are OH is the polar group. That's an alcohol like you would see in water or alcohols. Some of them have nitrogen groups like amino groups and whatnot. And you can see there's a few more there. Some of the amino acids are actually charged. And so we can group them into the negative or positively charged amino acids, which are sometimes called acidic or basic, based on their properties. So for example, we have glutamic acid here, and you can see there's that carboxylic acid side chain. And so 20 different amino acids, we have a huge variety in chemical properties. And so proteins can do a huge variety of biological functions because of that, because you can give them all sorts of different amino acids. So before I go into the notes, I just wanted to show you what happens when you attach amino acids together. You can have a dipeptide or a tripeptide. So if you take a look at here, we have 2 amino acids are attached, and we have another amino acid over here. And here is that dehydration condensation synthesis, where you're removing an OH and an H, making an H2O. And what you're doing is forming something called a peptide bond. And so now we're making a peptide. So maybe now I'll make a few of those notes and summarize them in that Word document. Let me just pull that up. Where is it? Right there. So you can see just basically picking up where we left off, except for now we're talking about proteins, which are sometimes called peptides or polypeptides, of course. Poly means many, many amino acids, of course. So the basic unit is an amino acid or amino acids. The chemistry, they have carbon, hydrogen, oxygen, and nitrogen. So we're looking at COOH, or sometimes that's COO with a negative charge on it group, and NH2, or sometimes NH3, and the NH3 has a positive charge. So what I'll do is just maybe I will fix that for you so that it looks a little bit more like the proper chemistry. There we go. And 3, of course, is a little 3, and the plus is, of course, a superscript. So NH2. There we go. What are the linkages called? They are called peptide bonds. And what do proteins do? A whole bunch of things. So let's see if I can remember what some of the things we talked about were. We have enzymes. We have receptors. We have antibodies, membrane, transport proteins. We have structural proteins like collagen. I feel like there were a couple others. I'm just struggling to remember what they all were. We have muscle tissue. So maybe I will just put etc. A whole bunch of different functions. The big one, of course, we're going to talk about in this class is enzymes, and the other big one is membrane transport proteins. So we do have a couple more things to say about proteins before we get to the nucleic acids. Somebody's mentioning transmitters. I imagine you mean, do you mean neurotransmitters? Is that what you're talking about? Some neurotransmitters are not proteins. There's actually a variety of things that we call, I guess, hormones and transmitters. Some of them are protein-based and some of them are not. But good point. Okay, so back to our PowerPoint. Like I said, a few more things to say about proteins before we move on. One thing we are going to talk about later on in the course is translation. Translation is the process of protein synthesis, and that's what happens at the ribosome. You can see kind of a summary of the process there. This is where the messenger RNA, which has the nucleic acid sequence on it, is being read at the ribosome and converted into an amino acid sequence or a protein chain. So more on translation a little bit later on in the semester, in the last kind of third of the semester. Another thing to say about proteins is that their structure is relatively complex compared to a lot of the other biological molecules. And sometimes that complexity is talked about in different levels of structure. So the primary structure is just the sequence of amino acids. So you can see here's a very short peptide of 25 amino acids. So you can see glycine, proline, threonine, glycine, threonine, and so on. And so what happens is that this protein is going to be made at the ribosome. And as those amino acids start to get strung together, they start to form something called secondary structure. So the secondary structure is where all of those polar groups start to interact with one another. And they're polar, right? So they have partial charges. And so they actually start to form a lot of hydrogen bonds. And there's a few different structures they can form. The most common structures by far are these two, the alpha helices. So that's shown here on the top. And literally the strand starts to form a little twirl or a helix. And you can see the hydrogen bonding here is in between. And this is a really, really common structure you see in proteins, probably the most common type of secondary structure. Sometimes they form these sheets. They're called beta sheets or beta pleated sheets. And this is where there's hydrogen bonding between the different strands. And so the sheets are pleated, meaning there's a little bit of a zigzag to them in terms of how they fit together a little bit better. And whether you get an alpha helix or a beta sheet kind of often depends on which amino acids you have in terms of how they're going to fit into the structures. So they come off the ribosome, they've got this amino acid sequence, they start interacting, forming these hydrogen bonds, and there are other chemical forces that may lead to that final protein structure. So all the usual chemical forces that you've talked about in introductory chemistry, such as other dipole interactions or hydrophobic and London dispersion forces and ionic bonds. And eventually you get what's called kind of the final folded structure, which is called the tertiary structure. And so this is just representing some of those other forces. And your final protein might look something like this. So kind of like a blob. You can see on the far left is what is called a space-filled model. And it's trying to show all of the atoms in that protein. Actually, it's not even showing the hydrogen atoms. The black or gray is carbon, red is oxygen, yellow is sulfur, blue is nitrogen. And it's a little bit messy. You can sort of see the active site. The active site is this opening here or this cleft. That's where the substrate is going to react in it. But what we often do, we don't often draw these. It's just really messy. And so what often is done is sometimes on the right-hand side is something called a wireframe, which is trying to simplify this thing on the right. It's also a little messy. So usually proteins are kind of drawn like the structure in the middle. It's called a ribbon model. You can see the alpha helix is done with these little swirlies here. And you can sort of get a better idea of the fold and how the structure of the protein is done there. And the beta sheets are represented usually by little arrows or little bands like this shown on the right here. So that's kind of how proteins form together. Now, there's actually one more level of structure called the quaternary structure. So some proteins actually complex with other proteins to make kind of what we call a larger protein or a quaternary protein. So a fourth level of structure. And I'll show you a couple of examples here. We already talked about collagen. Collagen is like I said, is rope-like. It's found in your skin, and it's actually 3 peptide chains wrapped around each other, just like a rope. And of course, that's what it's being used as, kind of in your skin. It's a structural thing. It's giving you, like I said, giving your skin some elasticity and some, a little bit of flex to it, right? Another one there on the right is hemoglobin, and it's actually 4 peptides together. And so one hemoglobin will actually carry 4 irons, which means it will carry 4 oxygen molecules around in your blood. So you see this quite often with proteins, this quaternary structure. Not every protein, but pretty common. Sometimes you even see proteins that have, these ones here, you can see the subunits are identical or almost identical. Sometimes you see proteins like this. This is ATP synthase. We'll talk about it as well in topic 10. And you can see it's actually made of multiple subunits, and they're not all identical. There's like 15 subunits or something like that, and there's actually quite a few different ones. So more on ATP synthase later on in the semester. So one more thing to say about protein structure, and we're kind of going to talk about this just a tiny bit over the semester, is that sometimes things go wrong. And so this is actually the cause of a few human diseases. And I want to talk a little bit about sickle cell anemia today, and we'll talk about it a couple more times over the semester. But these are diseases that are related to misfolding of proteins. And you can see there's some other ones listed there, like Parkinson's or Alzheimer's as well. And some of these are mutations in our genome. Sometimes there's other causes to them as well. We don't fully understand Alzheimer's and Parkinson's, the causes of those diseases, but they are due to proteins misfolding and aggregating in those cases in the brain, unfortunately. So let's just take a quick look at sickle cell anemia. I want to show you what is going on there. So if you take a look, we have our normal protein here. It's a hemoglobin, and you see the amino acid sequence. And down here on the bottom, number 6 is different. So number 6 in the normal hemoglobin is a glutamic acid, and number 6 in the sickle cell anemia is a valine. So glutamic acid is polar and charged. Valine is hydrophobic, very different chemical properties. So let's just take a look. They show you what's going on with the structure. So there's our normal hemoglobin. As I mentioned, it's a quaternary protein. It's got 4 subunits. And it sits around in the red blood cells, carries oxygen, and that's kind of the normal thing that is going on. With the mutated protein, this is a genetically inherited disease. The protein shape is a little bit different, and it's got that hydrophobic component to it. And so what happens is in the cell, the proteins, they clump together. And they can still carry oxygen, just not as efficiently. And unfortunately, this clumping together forms these fibers, not very flexible fibers. And so it turns out that the cells kind of look like that. So you're probably thinking, okay, what's a sickle? A sickle is like a tool. It's like a stick with a big blade on the end, and it'd be used to thrash and maybe harvest wheat or something like that. It's also the kind of thing a worm reaper would have. And so that's where the name of the disease comes. Anemia means you don't have enough red blood cells. And so these red blood cells, actually, they're very fragile and they break. And so people with sickle cell anemia can have some serious health issues. And like I said, it's a genetically carried disorder. And we're going to come back to it later on in the course. I think much later. I think topic 15 is when we come back to talking a little bit more about sickle cell anemia and mutations and things like that. Okay, I think that brings us to here. So there's our little cell. It's got membranes to keep in the water. It's got some food. It has a cell wall. And now with proteins, it can actually do stuff. It has enzymes. It can do reactions and a whole bunch of other things. Last part of this formula, of course, is the genetic information. And that is where the nucleic acids are going to come in. So there's my reminder to make a table, and we've been working And we're going to talk about nucleic acids now. Oops, okay, looks like I got a repeat here in my PowerPoint. Sorry about that. Okay, let's talk about nucleic acids. So nucleic acids are, the subunits are nucleotides. So this here is a nucleotide. So take a good look at that nucleotide there. We actually have, where's my cursor? Oh, there it is. Right here in the middle, we have a sugar. So nucleotides have sugars in them. We call this sugar a pentose sugar because it has 5 carbons in it. And so you could have ribose or deoxyribose. And I might have a picture of those in a minute. If not, we'll talk about them later on in the semester. So nucleotide is made out of sugar. It's made out of a phosphate group, which is there shown on the left. The last time we saw phosphates was with phospholipids, of course. And we have a nitrogenous base. And I think I have the nitrogenous bases here. There we go. And if you take a look at the nitrogenous bases, they're called nitrogenous because they have a whole bunch of nitrogens. That's where the name comes from. They've got a bunch of nitrogens. So there is, of course, the different nitrogenous bases. You're probably familiar with them a little bit, the letters of the genetic code. So the T, C, T, A, and G. T, which is for thymine, is found in DNA. And then there's that 5th one, uracil, which is found in RNA. And like I said, we'll talk a bit more about these later on in the semester, starting around topic 13 or 14. We'll get more into DNA and RNA and things like that. There's the 2 sugars. We have deoxyribose and ribose. So if you take a look right here, carbon number 2, if I look on the RNA, there's an OH. If I look on the DNA, there's an H. And so literally deoxyribose is missing an oxygen. That's where deoxy comes from. So there's DNA, probably the most important, or at least the one we talk about the most, nucleic acid. And DNA is a double-stranded molecule and also forms a helix. Don't get that confused with the alpha helix of the proteins. This is a double helix. It's a little bit of a different structure. And we've got all that base pair matching going on in the middle. And those are matched together with hydrogen bonds. So A matches with T and G matches with C and so on. And then there's the structure of the double helix shown in a kind of a stick or a wire frame model. So DNA, of course, is the storage of genetic information. And it's kind of the perma storage where we put our information on how to build a new cell. I think I have a picture of RNA here. I meant to put a picture of RNA as well. RNA is related to DNA. It's usually single-stranded, and it carries information in more of a temporary state, right? It's bringing the information from the DNA. It's going to bring it over to the ribosome to make the proteins. So much more on nucleic acids, like I said, later on in the semester. One other nucleic acid I want to show you is not dealing with DNA. Here's another nucleic acid. This is ATP. So ATP, take a look at this. It's a nucleotide. We've got a pentose sugar here. We've got a nitrogenous base. The A actually stands for adenine. And we have, in this case, 3 phosphates. So ATP, of course, is involved in energy transmission in a cell. So those are kind of the 2 prongs of nucleotides, right? They're either involved in information storage or transmission or energy transmission. So we'll be talking a lot about those things later on in the semester. So maybe now is a good time to go back to our notes here and put in some details. So nucleic acids, of course, are made of nucleotides. And nucleotides contain a phosphate, a pentose sugar, and a nitrogenous base. Okay, maybe what I'll do, I'm just trying to think of a way to not make this so messy. It just likes to go on to the next page. Okay, the chemistry. So we have carbon, we have hydrogen, we have oxygen, we have nitrogen, and we have phosphorus in the phosphates. Okay, so I'm not going to go into any more of the details on that. Hopefully you can recognize them, but it's got an extra element in there. The bonds, I didn't actually talk about them, but they're called Phosphodiester bonds. I won't ask you about bonds on a test, by the way. I might say the words in lecture, which is why I'm putting them there, but I won't directly test you on which bond is found in a carbohydrate versus a lipid or something like that. But if you are into chemistry, then sometimes that language does help you understand what we're looking at here. So we're looking at information storage, that would be DNA, or transmission, that would be RNA. And the other function, of course, is energy. We'll call it transmission, meaning energy is being transmitted between, let's say, two organelles or from one chemical pathway to another, and that would involve ATP. And there are other nucleotides, such as GTP, which we will see this semester. All right, so I'm hoping everybody got all that. I'm going to go back to the PowerPoint in a second. And like I said, if you don't get all this, you can always review the lecture recording, or you can ask me for a copy of this. It's not really a big deal for me to just email you if you want a copy of my Word document. Okay, a couple more small points about DNA and RNA. This picture here, we're going to see it quite a few times in the last part of the course when we start talking about DNA and RNA and proteins. And so these processes, replication, transcription, translation, we're going to talk about them a lot. So it's just good to throw the words down now so that hopefully we can start recognizing them, thinking about them a little bit later in the last part of the course. And one other thing we are going to talk about a lot in this course, of course, is the difference between eukaryote and prokaryote cells. So bacteria are prokaryotes, they have no nucleus. And so all of these processes, replication, transcription, translation, they all happen in the cytoplasm because that's the only place for them to happen. In eukaryotic cells, the DNA stuff, so replication and transcription take place in the nucleus, and the ribosomes are found out in the cytoplasm, and so translation happens out in the cytoplasm. But more on this later. So a couple more things, and then I have a Kahoot for you. You might see sometimes these words used combined with other words. You don't just have to have a lipid alone. You could have a lipid connected to a protein. So that particular molecule would be called a lipoprotein. You might also have something that has a glyco in it. So the process of adding a carbohydrate is called glycosylation. So you could have a glycosylated protein or a glycoprotein, and that would be a protein with carbohydrate added to it. On the other hand, you could have a large carbohydrate with a small protein added onto it, and that's something called a proteoglycan. So if you ever see these words, right, like I said, it's like this is kind of how the English language works. Sometimes we mix and match things, and you end up with something that's new, and these would be considered complex or conjugated macromolecules. So there's our little cell. It has a brain. It's remembering what to do, and it's all ready to go. And here is our, I guess there's the proteins again. And then here's kind of the breakdown in terms of the percentage of, by mass of what you're looking at in a cell. So lots of water. Proteins are kind of the big one, followed by RNA, phospholipids, of course, because of the membranes. And then a small percentage is actually polysaccharides and DNA. That doesn't mean they're not important, just in terms of the mass, that's what we're looking at. So here we are at the Kahoot. So load up your app and let's do this. I'm gonna pull that up here. And here we go. I guess I have to load it. So just give me a second here. For some reason, I thought I had it loaded. You can see I have a whole bunch of them there. Okay, there we go. So we are, hit start here, start. And macromolecules part 2. There we go. All right, get ready for the pin number. Here it comes. It's 2890834. All right, I'll give you guys another 15 seconds to join. All right, just 3 people for now. Let's get going. It's worth it to take a look at this one to try to understand some of the differences between the macromolecules. Very important for this class. So what category of macromolecule does this belong to? We're timing out here. Got to think quick. Let's take a look at this. This here is actually cholesterol, or no, sorry, this is testosterone. So this is a steroid, and you can see the 4 rings, and that makes it a lipid. Question 2, which category of macromolecule does this belong to? All right, timed out again. This is actually an amino acid. Take a look. There's the amino group. There is the acid group. And so that makes this one basically part of the protein category. What category of macromolecule does this belong to? So this is a nitrogenous base. You can see all those nitrogens here. This is, I think it's an adenine, if I remember correctly. Question 4, what category of macromolecule does this belong to? So this here is a lipid. You take a look at this, it is a whole bunch of carbons and hydrogens. This is actually a fatty acid. Question 5, what about this one? Nucleic acid, good work. So maybe recognizing all the G's, U's, C's, and A's. That is actually transfer RNA. All right, 3 more questions. What about this one? This is a carbohydrate, so good job. This is actually sucrose. You can see all of those OHs in there make this a carbohydrate. Question 7, 2 more to go. This is a protein. So look at all those ribbons on there. That's usually how we draw proteins in textbooks because we're not trying to draw all of the atoms. All right, last one. So this here is a nucleic acid. Take a look. There's the phosphate, the pentose sugar, and the nitrogenous base. So we can take a quick look at the podium, not getting a lot of participation on this, so we can, hopefully we can do better next time. Part of the learning process is to participate, and so just a reminder on that, and it's important that you know how to recognize these molecules, because on your midterm, you are going to have a question, a matching question that asks you to do just that. So somebody's saying, can you raise the time? Maybe next time, we'll see. I have to basically go through the quizzes and try to decide how much time I need on each question, but kind of intentionally want to make that one a little fast just to get people so I could ask more questions. I just want to show you on the one other tool to help you studying on the Moodle page here. Let me just actually change this to student view so we can make sure it looks proper for you guys. So there's the Moodle page, right? And you go down past the notes. I'm going to be adding some quizzes here. The quizzes are not for marks. The quizzes are just for review purposes only. But hopefully you can make use of them as we go through the topics. It gives you a little bit of an extra study tool so that when you get to the midterm, you've had some practice. All these are multiple choice. The midterm will have some written components and multiple choice. But like I said, the quizzes here are kind of designed to, help get you familiar with some of the questions, because you know what, the test can be a lot trickier than a lot of people, I think, anticipate when they first see one of the biology tests. All right, so I just loaded up the wrong PowerPoint. So that is it actually for topic 2. I'm gonna just exit that PowerPoint, and we've got a few minutes left, which is just about right. What I want to do is kind of introduce the next topic. I'm just loading it up here. Just give me a second. And then we get a chance to talk about it next time. So the next topic we're doing is topic 20. And I told you this when we were going over the course outline that we would be bumping topic 20 to the beginning of the semester. Topic 20, you can see, is on viruses, and there's content there about the new SARS-CoV-2 virus, and also a little bit of information on vaccines. So very relevant information. I'm hoping you take some of this information and tell others about it. So I just want to introduce viruses for today, and kind of talk about a little bit about what they are. So often I start off discussion in class and say, hey, has anyone here ever had a viral infection? And I think most people realize that, yeah, they've had one. Whether it's COVID-19 or something else like that. Then I throw this one up here. You can see this is a herpes virus, and ask if anyone knows if they've had this one. And one or two people might recognize it. A lot of people, of course, are like, okay, I'm not going to stick my hand up if I've had herpes. But I'm here to tell you that actually herpes virus infections are super common, and every human on the planet has probably had at least one, probably 3 or 4. Herpes simplex 1, you can see, is up here, and there's actually 2 herpes simplex viruses, 1 and 2. Herpes simplex 1 is the one that causes cold sores, and so about 50 to 60% of Canadians have cold sores. And these are little blisters that sometimes happen around often on the face, around the lips, and they're very, very, very common. And it's a lifelong infection. With all herpes viruses, you get infected and it's lifelong. Herpes simplex 2 is more commonly associated with a sexually transmitted genital herpes, and that's maybe 10 to 20% of Canadians. The numbers are not perfect, but that's kind of a best estimate. So if you have a chance, then you may have one of these herpes viruses. If not, don't worry, there are more herpes viruses. There's a 95% chance that you have been exposed to chickenpox. That's caused by the varicella virus, or 95% chance by the time you're age 30 that you've had Epstein-Barr virus that causes mono or infectious mononucleosis. So these things are pretty common, and these are also lifelong infections, and which means you have them for life. If you haven't had any of those ones, then there's a chance that maybe you've had one of these herpes virus infections. You can see the rosy yellow virus is about 100% of humans have that. I think by the time you're 6 months old. you're likely infected with it, and it is lifelong. So viral infections are really common. And kind of part of this is to say that it's not necessarily something to be scared of. Most of these diseases are not life-threatening. And in fact, a bunch of these, the ones I currently have highlighted, most of these people have them and they don't even know they have. They're infected, and that's kind of it. Sometimes when you are immunocompromised, you might have symptoms, but for the most part, most people don't even know they've had them. There's one more herpes virus. I'm assuming none of you have had it. It is called the B virus, herpes B virus. It is 100% fatal. So that's why I'm assuming nobody here has had it. So a few more things I want to say about viruses. Viruses, as far as we know, they seem to infect every living thing on the planet. Wherever we look, whether it's in frogs or flowers, or humans or elephants or bacteria, all of these things seem to have at least one virus that infects them. So they're very, very common. And we are exposed to billions of viruses every day. I'm showing you that picture because if you had a salad today or lettuce on a sandwich, then you have probably eaten viruses that are on that have either infected the plant or infected the insects that walked on that plant when it was back in the field. We breathe them through our mouth. Our bacteria in our gut, there's E. coli. E. coli lives in your intestine. It is infected by its own viruses. So most viruses, the reality is, have little impact on human health. Partly it's because they're plant viruses or viruses infect other things like insects. Partly because our immune systems are really quite remarkable. There's only really a couple hundred of them that actually infect humans, compared to the potentially billions and billions of different varieties of viruses that are on the planet. So what is a virus? Here's a definition. This is from, I think, one of the textbooks I have on my shelf, and it describes them as minuscule, so it's very small. Acellular, remember we had talked about the different types of cells, prokaryote and eukaryote cells. We're gonna get into the details in prokaryotes and eukaryotes a lot more in the next few units, but these don't have cells. So it says it's basically a nucleic acid surrounded by a protein coat. So picture it, a nucleic acid is instructions, and it's packaged up with a little bit of protein. And what the instructions do is the instructions say, make more viruses. And that's it. And unfortunately, some of them can cause some issues. And I'll show you what some of these viruses look like. The other thing I want to show you is what small looks like. So there's a human that's actually a pretty tall human at 2 meters, but just to make the math easy. And there's a virus about 200 nanometers. So that would be the size of a rhinovirus that causes a cold. And if you look at the scale, here's the math, right? I'm converting everything to nanometers. So that's 2 billion nanometers. And so we're about 10 million times larger than your typical virus. If that math doesn't help make sense, I'll show you another way to look at it. So there's a pin, and that's about 2 millimeters or 2,000 micrometers. A dust mite is about 300 micrometers. That's just barely visible. And a strand of hair, these are two things that are barely visible by the human eyeball. Just within that range of they look like a speck. A red blood cell is about 10 times smaller than the width of a strand of hair. So it's 10 micrometers. So let's zoom in to a rhinovirus. A rhinovirus compared to a red blood cell looks like a speck. It's so small you can only see it with an electron microscope. And we're going to talk about electron microscopes in topic 3. So we'll come up to talking about those in a minute. So very, very tiny. So what does this mean? It means that on that pin, we could fit about 500 million rhinoviruses. And yeah, so one sneeze, you could in theory infect thousands of people with one sneeze. They're very, very, very tiny. There's another way to look at this is all of the coronavirus infections on the planet could basically fit into one can of soda, such as a Coke can or something like that. So very, very, very small. Another way to look at them is the size of their genomes, and I'll show you some pictures of a few of them before we wrap up today, a few more viruses. If you look at a human cell, there's some numbers of the size of a human cell, and the number of genes is about 20,000 or so. A bacteria is about 4,000 to 6,000 genes, and a virus is anywhere from maybe about 3 to 50, or some pretty common numbers. So very, very tiny compared to humans. There's a picture I showed you before showing the two different types of cells, the eukaryotic cell on the left, the animal cell, the prokaryotic cell on the right, the bacterial cell, and then a virus looking like a speck compared to E. coli. Just zooming in on that, you can see that the virus has some genetic material. It can be DNA or RNA. So that's something we're going to talk about next day. And this particular virus here actually has a membrane as well, which is not found on all viruses. I'm going to skip this one for now. I want to show you some pictures of some different types of viruses and their structures, and then kind of I'll come back to that next day. So here is your basic virus structure. You've got some genetic instructions, and the genetic instructions can be DNA or RNA. They can be, and actually you can have double-stranded DNA or single-stranded DNA. You can also have double-stranded RNA or single-stranded RNA. So kind of a little bit different than what you'd see in cells. We don't usually see double-stranded RNA in cells, so that's kind of weird, but viruses are weird in general. The virus is packaged up with a protein coat, and that protein packaging has its name, and it's called a capsid. And I will review these terms when we come back next day. Now, sometimes a virus might grab the membrane of its host cell on the way out, and this we actually call an envelope, and often these ones will have these viral glycoproteins, which we often call spike proteins. So that's what we're talking about a lot when we talk about the coronavirus, so these spike proteins. And that's your basic virus structure. I'll show you some pictures of a few, and then we'll wrap it up for today. Here's some different viruses. They come in many shapes and sizes. Some of them are kind of helical. Some of them are more geometrical. Some of them have membranes. Some of them do not. You can see they all have a genome. They all have the protein packaging. There are just different ways to do it. That's where I'm going to wrap up today. We're going to come back next day, talk a little bit more about virus structure. We're going to talk a little bit about how they actually replicate, and we'll probably get into and start talking about coronaviruses and COVID-19. And that will probably be Monday's lecture.